In this video, I want to discuss creatine, beginning with the synthesis of creatine specifically. An overview of the biosynthetic pathway of creatine is shown here. As you can see, creatine is synthesized de novo from arginine and glycine. These amino acids undergo two reactions catalyzed by, first, L-arginine glycine amidinotransferase, and second by guanidino acetate methyltransferase. The end products are, of course, creatine, but also S-adenosylhomocysteine, a process I will discuss now in more detail. The first reaction catalyzed by arginine glycine amidinotransferase occurs as follows. The first step utilizes the thiol group of the cysteine 407 residue. This residue harbors three functions, specifically action as a nucleophile, acid, and a base. As shown, there is a nucleophilic attack of the carbon atom of the amidino group of L-arginine, while cysteine 407 also protonates the epsilon imino group of the arginine leaving group. Following this, the bond between the epsilon imino group and the amidino carbon atom is broken, forming L-ornithine. L-ornithine then leaves the active site of the enzyme, permitting the entrance of glycine, which then undergoes deprotonation by the histidine 303 residue. This leads to a lone pair of electrons on the nitrogen atom of glycine, facilitating a nucleophilic attack on the amidino carbon atom of the cysteine 407 residue. In the last steps catalyzed by this enzyme, the amidino group is transferred to the glycine and in turn is removed from cysteine 407, thus forming guanidino acetate and restoring cysteine 407. This is an important step as, if you recall, enzymes may ephemerally be altered during a reaction but are not permanently changed from catalyzing a reaction. The pseudo-exception to this rule would be, for instance, suicide inhibition, where an enzyme reaction with a substrate irreversibly inhibits its action. Moving on to the second reaction catalyzed by guanidino acetate and methyltransferase. This begins with a simple proton abstraction from guanidino acetate by the aspartic acid 134 residue. This results in a negatively charged nitrogen atom shown here, as it now has an excess electron over its desired number of valence electrons, being 5. Next is an important function of S-adenosylmethionine, or SAM. For the unaware, SAM is a widely used methyl group donor. As such, guanidino acetate undergoes methylation by action of the electrons on the nitrogen that was subjected to the proton abstraction. As a result of the loss of the methyl group, SAM becomes S-adenosylhomocysteine, and importantly, creatine is formed. The resultant creatine is then able to function in the phosphocreatine energy transduction system. In this system, creatine is able to become phosphorylated within the mitochondria and form phosphocreatine, which then may egress to the cytosol and donate the phosphate group once again to resident ADP and form ATP for further energy transfer for a plethora of cellular processes necessary for survival. However, limited creatine synthesis occurs endogenously, leading to the widely used creatine supplementation that occurs today to potentiate the bioenergetic impact of creatine, especially in the context of exercise, as muscle action is a highly energy-demanding process.